Hello, class. So today we're going to basically be covering cell membranes, the secretory pathway, and transport into the nucleus. The first part should be all review for you guys, especially anybody who's taken basic biology. Cellular membranes are a big part of cells, and we'll cover that, and then we'll get into the secretory and transport into the nucleus in the second part of today's lecture. Um, so, as I'm sure you guys all know, eukaryotic cells, they have individual compartments, and those compartments are actually called organelles. And those organelles actually carry out different functions. And we'll look over the course of the next few lectures at what the different organelles are doing. In certain instances, we've already seen, what happens in the nucleus and things like that. But what separates these compartments are lipid bilayers. And it's the lipid bilayers that really give and allow the compartments to have different chemical compositions, both in proteins and in their biochemical functions. But because the cell is actually compartmentalized, proteins and small molecules need to move in and out of these different compartments and move to those compartments and get sorted to the appropriate compartments so that they can do their job. Okay, and these compartments that are composed of lipid bilayers, basically, they have different membranes, and in the different membranes there's different lipids and also different proteins, and that really helps give the different organelles their specific function. And if you guys want sort of an example, if you sort of think about the chemical composition of the mitochondria, you actually have multiple compartments in that one organelle. You have an inner mitochondrial membrane, and that's going to be very different from the proteins and the lipids that are on, for example, the plasma membrane. So just a quick overview. Here's a great little image of a cell, and you can actually see the lipid bilayer right here. And if you were to sort of sketch it out and draw it into a schematic, what you have here are the lipid molecules, and then also you can have proteins embedded in the lipid bilayer. Okay, and these have transmembrane spanning oftentimes alpha helices, and we've seen some of these in the case with G protein coupled receptors, as well as receptor tyrosine kinases and other proteins that are in the lipids. But they can actually span the lipid bilayers, and we'll cover more about proteins that are basically put in the lipid bilayer in a couple lectures, but I think this really gives you an idea. One of the things you have to know about the lipids are they're amphiphilic, okay? And what that means is that they have a hydrophilic head group and a hydrophobic tail group. These two little legs that are coming off is the hydrophobic tail, and the red sort of egg shape here is actually the hydrophilic head group, okay? And if you want to look chemically at the different phospholipids, they're all relatively similar in a lot of ways. In this instance, we're looking at phosphoglyceride here, and you have the glycerol, then a phosphate group, and then a choline, and then you have your hydrocarbon tail. Okay, so this is the polar hydrophilic head group, and then you have sort of a non-polar hydrophobic tails. And these hydrophobic tails can be either saturated or unsaturated. Okay, so this is an unsaturated one where you're going to have a double bond, and that creates a little kink in it, as you can see here. Okay, and so one of the things that you do need to know is that most phospholipids, they sort of have this 3-carbon glycerol backbone, and then and in the case with phosphoglyceride, you have the phosphate and also the choline group. Here's just an image of some abundant phospholipids. You have phosphatidylethanolamine here, phosphatidylserine, phosphatidylcholine, sphingomyelin, and sphingosine down here. You guys don't necessarily need to memorize the chemical structure or how these things differ, but you should be able to recognize what what a lipid molecule is based on its name, especially these ones. And so just for general purposes too, it turns out that A, B, C, and D here, these are actually the most abundant phospholipids that are in a mammalian cell, okay, and they represent greater than 50% of all the phospholipids in a cell, right? And these will make up the lipid bilayer of all the different compartments in the plasma membrane and whatnot. And that includes like the ER, the Golgi, and the mitochondria and things like that. Another molecule that you need to know about when talking about the lipid bilayer is cholesterol, because this actually gets embedded in the lipid bilayer. Okay, and cholesterol, it's actually called ergosterol in yeast, which is it's a little bit different but pretty similar. It's a sterol that's basically very abundant, and one of the things that it has is this OH polar head group on the end, and then it has the sterol ring structure that's here, and then you have your sort of non-polar hydrocarbon tail. And so here's just another little schematic of it, and here's sort of a space-filling molecule. And we'll talk about lipid rafts in a second. We saw those in the video that we watched on the first day of class. But you know, lipid rafts, they actually are enriched with cholesterol, and it helps provide sort of tighter packing. Because here's how cholesterol actually fits in the bilayer. You have your polar head group along with the polar head groups of your phospholipids. And then the cholesterol actually will stiffen this region here, and then you have a more fluid region, especially if you have an unsaturated hydrocarbon tail that comes off. Now, something that you really have to appreciate with phospholipids, and this has to do with their amphiphilic nature, is the idea of polar versus nonpolar interactions. And we've sort of talked about this before when we were going over the biochemical section. But if you sit here and take a molecule like acetone and you put it with water, these both have a dipole. And so when you sort of mix them with water, the water can actually surround the acetone pretty well. But if you take something like 2-methylpropane, which is very hydrophobic, and you mix it with water, it's not going to sort of fit in the water as well. Okay, and so if you were to have a whole bunch of methylpropanes here, they would all sort of coalesce together and in the water and sort of separate, and so there'd be a phase separation here. And so it's the physical chemistry of the lipid bilayers, and it's, you know, sort of the combination of hydrophobic and hydrophilic interactions that are occurring in these amphiphilic lipids that sort of give them their character, right? And so to sort of look at that in a schematic representation, you know, if you were to just sort of draw out the shape of a lipid molecule, you know, it would want to pack so that the hydrophobic tails are all on the inside and the hydrophilic head groups are all on the outside. And what you would end up getting is sort of this lipid micelle here, where it's all packed together, and then water would be on the outside with the hydrophilic head groups. Okay, when you have two hydrocarbon tails off of it, it makes them more sort of planar, and they arrange in a lipid bilayer, such as is shown here. Okay, so just in general, the hydrophilic polar head groups are going to be exposed to water, and then the hydrophobic tails will all pack together. Another important concept that you guys all need to know is that lipids do not want to be planar. Okay, this is a very energetically unfavorable state because this sort of hydrophobic portion on the sides here does not want to be solubilized in water. And so because of this all these hydrophobic tails that are sort of sticking out in the amphiphilic lipids, they're going to basically come together and form sort of a sealed compartment because it's just thermodynamically unstable to be in this state here. Okay, so you get these sort of sealed compartments, and you know, now you can start to think about, you know, how mitochondria are set up and how the endoplasmic reticulum and lysosomes and endosomes and the Golgi apparatus, they're all going to be sealed compartments with an inside and an outside. And to give you guys a sort of a better idea of that, you can actually generate these spheres in a lab called liposomes, where you basically take phospholipids and you sort of mix them together, and they're going to make these spherical, empty sort of balls of lipids, where you're going to have water on the outside and water on the inside, and then your lipid bilayer in the middle. And these can be easily created in the lab, and there's a lot of interest in the pharmaceutical industry right now because it's thought that these could actually serve as sort of a great mechanism for targeted drug delivery. And that's really important to the pharmaceutical industry. And although I can't tell you exactly what are in the Pfizer and Moderna COVID-19 vaccines, they do contain some sort of lipid component, and it could be that the mRNA for those various vaccines are actually packaged in a liposome similar to what you're seeing here. Not identical, but somewhat similar. And then this delivers it to the cells. Now, a great way to study lipid bilayers is you use these things called black membranes, okay, or where you basically create a lipid bilayer between two pools of water, and then you can actually study the physical chemical properties of these phospholipids. Okay, and what they've sort of figured out just from studying this is that lateral diffusion is pretty dynamic, right? It's easy for the various lipids to sort of travel in and around in a single bilayer, but flipping is pretty energetically unfavorable, and if you stop and think about this hydrophilic head group having to pass through this hydrophobic portion and then get to the other side, that's actually a fairly energetically unfavorable event. And the way they sort of measure this is they can use spin labeling to measure the dynamics and follow how the various phospholipids are moving in these black membranes. You can also simulate it on a computer. So they have computer models, and here you have basically a simulation of your phospholipids. Here they are, and it's basically 1,000 simulated phospholipids in the 300 picosecond time scale here. And one of the things that they found is rotation and lateral diffusion is pretty common, but flipping of the lipids is actually rare. We'll talk about this more when we get to the endoplasmic reticulum, because most of the phospholipids are actually made on the cytoplasmic face of the endoplasmic reticulum, and then they flip, and there's actually an ABC transporter that actually helps to flip the lipids. So there's proteins involved that actually move them, but in and of themselves they won't typically flip on their own. Okay. Another thing that you need to think about is saturated versus unsaturated lipids. Okay, so the unsaturated ones are going to actually have a kink, right? There'll be a double bond in them somewhere, and so they sort of kick out. And this makes them sort of shorter and more fluid. Okay, so they can actually move around a lot more. There's less packing. When you get to the sort of the saturated hydrocarbon tails, there is no double bonds in them, and it makes the lipids a lot more sort of rigid and a lot less fluid. And cholesterol is typically embedded here along with the saturated ones, although you can have some here. And what ends up happening is these saturated phospholipids will sort of coalesce together, and they sort of come together and form these things called lipid rafts, which we'll talk about in a second. Okay, here's the typical lipid composition of a whole bunch of different cells here. You guys don't necessarily need to memorize all these, but I think it's kind of interesting for you to look at, because certain cells have, more cholesterol than other cells, and, we'll see in a second that, certain compartments in the cell will actually also have different phospholipids. Okay, so you can see that phosphatidylserine is relatively rare. Phosphatidylcholine and sphingomyelin are a lot richer, but you don't actually find any sphingomyelin in the inner or outer membranes of the mitochondria. So it's just an interesting thing to look at and know that the lipid composition is different depending on the cell membrane and depending on the cell type. Okay, now one of the things that lipids do is, and I sort of mentioned this before, the saturated and the unsaturated phospholipids are actually going to create subdomains. Okay, so if you basically take a liposome with phosphatidylcholine and sphingomyelin here, and you add in cholesterol, that'll actually separate them into subdomains all on their own. There's no proteins here. And this sort of is an indication that, you know, lipid rafts are actually favorable and can spontaneously self-assemble provided there's the necessary lipids that are composed in the cell. Okay. And here's just an EM of a synthetic lipid bilayer, and you can sort of see here where you're going to have the saturated phospholipids sort of coalescing, and the and the unsaturated ones will form a different area. Okay, and so these synthetic bilayers sort of really define and sort of prove the existence that lipid rafts can occur within the cell. Okay, and so what exactly is a lipid raft? Okay, lipid rafts are basically just subdomains within a lipid bilayer that tend to have fully saturated phospholipids with them, and then they actually pull in specific proteins. So what you end up having with, let's say, on your plasma membrane will be these domains where you have lipid rafts and then just the remainder of the lipid bilayer. Okay, and we sort of saw this also in the video on the first day of class that we watched, and if you guys go back and watch it, you'll see the lipid rafts sort of floating around. And the lipid rafts are often enriched in these proteins called caveoli, and they're involved in endocytosis. And you also have things like GPI-anchored proteins and other intercellular signaling proteins that can be membrane-anchored. Okay, another thing too is that, you know, oftentimes cells will store energy as fats in the form of triglycerols and cholesterol esters, and that actually happens in cells called adipocytes or fat cells. Okay, and so what ends up happening is they get stored sort of between the lipid bilayer, and this can actually be pinched off, and so then you have just this lipid droplet here where it's all hydrophobic in the middle, and it's got a single leaflet with proteins on the exterior of it. And fat actually is an amazing store of energy, and so your cells really like to do it. So not only do you have different phospholipids in different membranes, but the actual leaflets themselves will have different distribution of phospholipids. Okay, so there's sort of this asymmetric distribution. And so here would be outside a cell where you're going to have your phospholipids and your glycolipids, which we'll cover in a second here. These are shown with the blue hexagons here. And then you have your cytosolic lipids, which tend to be sort of negatively charged, like phosphatidylserine would be on the inside. And we've also seen other ones like phosphatidylinositol when we covered cell signaling, and we'll see that coming up in the next slide too. So in general, there's this asymmetric distribution. It's not only a compartmental distribution that different compartments have different lipids, but the leaflets themselves will have a different composition. And like I said, we sort of looked at inositol phospholipids when we were looking at cell signaling. So here you have your receptor tyrosine kinase. It gets phosphorylated. It will recruit an activated PI3 kinase, which then can phosphorylate the phosphatidylinositol 4,5 diphosphate. And then this phosphorylated inositol phospholipid can, serve as a docking site, or, it can actually be cleaved by diacyl or phospholipase C, and then go on to signal to the ER to release calcium, right? So in this case, you've got your G protein coupled receptor, you have your extracellular signal, and that basically will then activate phospholipase C, which will cleave off the IP3 here, okay? And then, as you guys remember from the cell signaling section, that IP3 stimulates the release of calcium. So here's the chemical structure of phosphatidylinositol 4,5 diphosphate. You have your inositol group, and then your phosphates, and then your glycerol here, and then your hydrocarbon tails on the protein. You can also get glycolipids, okay, and we saw those just a couple slides ago, right here, where you have these blue hexagons, right? These are just sugar moieties attached to a phospholipid, okay, and you know, you don't necessarily need to know these, but it's important to know that you can just get sugars added to them, and this happens in the interior lumen of the secretory pathway. It starts in the endoplasmic reticulum, and then the glycosylation gets further modified, and these sugar moieties get actually added to the phospholipids. And here's just the sialic acid group that's here in Nana that's there. And so these are oftentimes preferentially found in lipid rafts on the exterior of the cell surface. Right, and we'll see these again when we look later at different cell types and things like that. Okay, now as I've sort of really tried to drive home, the importance of this is that there's different compartments in the cell. Okay, and we saw this on the first day of class. Okay, and here are some of the cell compartments, and it's going to be important over the next few lectures to really know what's happening in all of these different compartments, right? You're going to need to know what's happening in the endosome, the peroxisome, the Golgi apparatus, the mitochondria, the endoplasmic reticulum. We've already looked heavily at what's going on in the nucleus with all the DNA and the chromatin, and then you also have the plasma membrane here, where you're going to have your receptors and other proteins that are associated with the plasma membrane. Okay, and we'll cover this a lot more over the next section, what these different compartments are doing. Okay, but here's just a good sort of representation of the different cellular compartments and how much of a cell they actually make up. Okay, so the cytosol is roughly around 54% of a liver cell, whereas the mitochondria represents 22%, and then you have your rough ER, smooth ER, and Golgi, and then your nucleus and some of the other components. Okay. And in those different membranes, you're actually going to have different liver or different lipids, and depending on the cell type, the amount of different membranes will differ. Okay, so if you look over here, you have a pancreatic endocrine cell. This is a sort of a heavy cell secretor. Secretes insulin as well as other things. It's going to have a lot of rough ER relative to what a liver hepatocyte has. Okay, but it also has then a lot less smooth ER, a little bit more Golgi. And so you guys don't need to know the difference here. It's just, I guess the point that I'm really trying to get across here is that different cells, depending on what they're used for in the body, will have different amounts of each of the different compartments. Okay, and you can sort of see that here in sort of this cross section of a liver cell, where here you have the nucleus, and then you're going to have your rough ER here, peroxisomes, and mitochondria, and then there's probably some Golgi and other things sort of mixed in here. Okay, so that's all I really have for part A. In part 2, we're going to look at transport into the nucleus, as well as some other stuff about the cellular compartments. Okay, so I'll be right back.